So good night. I think we have all the initial participants already in the meeting. I'd like to thank a lot Dushan for being here with us. I think this is a very important topic. It's always a crucial point when we have to explain a family, why different members in the family present with different manifestation of the genetic disease. And now we're having some light in this topic, and Dushan has been studying this a lot. So it's a pleasure for us to have you here with us, Dushan. We have some announcements here before we start the talk. So we would like to remind you that we are pretty close to our LASID meeting that will be held in Buenos Aires. You can point your camera to the QR code, and then you have all the information about the program, how to register for the meeting. And also we have now opened the call for cases. So if you have an interesting clinical case that you want to present during the educational day, you can submit this case up to August 20th, and the cases will be selected to be discussed during the educational day. Also, we would like to invite you to get to know and to read the papers that have been published in the new Journal of Human Immunology. It's a new platform that is dedicated to human immunology, and it's a very important, it's a great opportunity for us to publish our papers, since it's a journal that opens the opportunity for the different immunology societies, including LASID. And finally, please join LASID if you're not a member. So here you can find information how to join LASID as a regular member or a junior. So if you are below 35 years old or you are still in immunology training, you can join as a LASID junior member. And the registry is back online. So we have been facing some transitions, let's say, for the registry. And now we have it already set and online, so you can look for information about our statistics, general number of patients registered, and it's also an invitation for all of you to register in this important database for patients in Latin America. So Professor Dusan Bognovic, he's a professor of pediatric immunology in Columbia University. He's also the vice chair for basic research in pediatrics in Columbia University and Department of Pediatrics. And more than that, we have been neighbors in the lab, right? So our lab bench were not side by side, it was like back to back.
Back to back, yes.
Back to back. So it was like a really good time that we had in Jean Laurent's lab. And it's really nice to keep in touch and to be able to see how you were developing your work outside our former lab. So Dushan, thank you again, and we're looking forward to hear from you.
Well, thank you so much for this very kind invitation. I have also enjoyed, let me just start this up. Okay, you see me well? And let me just put the laser pointer. Okay, we are set. Thank you, Carolina. It's absolutely lovely to see you at the meetings and catch up and talk science and talk about patients. And then also because, you know, we sat back to back for quite some time in New York, not that long time ago. Happy early birthday. I know it's coming up for you, so.
Yes, thank you. In a few days, and then it's yours.
Correct. Okay, lovely. Thank you so much for taking the time to listen to what we've been playing with for past few years. And we are really talking about what Carolina said, and that's the concept of incomplete penetrance. I usually like to start with thanking people because I'm always petrified that I'll go over time or something like that and won't be able to do. So this project was started by an MD-PhD student in the lab, Connor Gruber, and it was really finished, and most of the work was done by another MD-PhD student in the lab, OJ Stewart, and it was helped by Haley Randolph and Meredith Ramba a lot, but actually with everybody else who's on this list and many collaborators without whom obviously none of this would be possible. So thank you everyone, truly. Okay, so I'm preaching to the choir here. You guys know extremely well what inborn errors of immunity are. It's genetic predisposition to infections, autoinflammation, autoimmunity, allergy, and malignancy. So I'm going to talk about, very important concept, which is what Carolina mentioned, and that is that incomplete penetrance is actually a very common ground in genetic disorders. And we don't necessarily know how to deal with that, right? Here is a made up family pedigree where where father carries the mutation and has a clinical disease, but there's three children, and all of them carry this dominantly inherited variation because the mother is wild type. But we see this in the practice that one of the children have early onset full disease. One child, another daughter here, could have a partial phenotype, and we call that's a form of incomplete penetrance, which we call variable disease expressivity. So not a full-blown disease. And then, of course, there's one child that carries it and is completely healthy. And it's a big problem because how do you manage this? Do you think that they will develop disease? Do you only treat the disease once it's developed? It's hard. Okay, so second concept I want to talk to you about is something that I had to relearn. So I wanted to apologize in case this is obvious to you. We are disomic individuals. We have a maternal allele copy of every gene and a paternal copy of every gene, and we are biallelically expressing them. There's two copies of DNA, same amount of RNA, same amount of protein, right? That's how I was taught biology, and that's how I was taught genetics. So now there are exceptions to these rules, and we all know about genomic imprinting, and that is a cluster of gene that have been well defined now that can be paternally or maternally silenced. So what that means in this example here, that every cell in our body is going to be paternally expressing, right? And those are imprinted genes. Another concept of monoallelic expression is X chromosome inactivation. So for everybody in the audience who has two X chromosome, in every cell there is a choice about which of the two copies is going to get inactivated, and that we know is happening from exist mechanism of X chromosome inactivation. Now there is another concept here, and that is one on transcriptional bursting. So transcriptional bursting is when a given cell starts for some reason transcribing off of one allele very strongly. But that same cell in some future time, 2-3 days from now, can actually start transcribing from a different allele. So you end up with a mix, right? So we won't be talking about any of these three concepts today. We'll be talking about this last one, which is called autosomal random monoallelic expression. And what it is that we are all born disomic, well, most of us are, and we have two copies of the genes. And then somewhere along development, it could be just in subset of T cells, B cells, in the skin or in the brain, we can have some cells somatically commit to the expression of the maternal allele or paternal allele, or biallelically expressing like it was meant when we were born. And you can think about this as transcriptional mosaicism, right? So your genetics is the same, but your transcriptional phenotype is different, right? Which is what this is. Okay, so now if we try and merge these two concepts of biallelic expression and monoallelic expression and incomplete penetrance. Typically, in this made-up family where everybody has a variation, but only the child is very sick, the father is partial disease and mother has no disease, we think about that gene expression as being equal in all three of these cases, right? But if we now shift our mind armed with monoallelic expression, we can pose a hypothesis that perhaps somebody who is wild type, I mean, somebody who doesn't have disease, is predominantly expressing the wild type allele. Somebody who has partial disease expressivity is more expressing the mutant allele, whereas that person with fully penetrant disease is actually preferentially committing to just the faulty allele, right? So when presented like this, I mean, it kind of makes sense, but what actually brought us here is the following. This was in 2017 that we have identified a patient that has a mutation in JAK1. And as you can see in this family pedigree, this is a de novo mutation. So this patient turned out to be genetically mosaic, with about 50% of the cells in the body being completely wild type and 50% of the cells carrying a heterozygous mutation. So the thing that I got excited about doing was single cell RNA sequencing, because I thought that this experiment of nature truly would be very informative, because in the blood we'll have half the cells that are completely wild type and the other half that carry the mutation at DNA level. So I wanted to see what would be transcriptional signatures that are intrinsic to the cells that carry this variant, right? Because we have a wild type control in that very human. So for what we did is, each dot here is essentially a single cell. And if you plot a random gene that has a variant allele and a wild type allele, basically as soon as you get to 10 transcripts, this is 10 to the 1 here. So if you're above 10 transcript depth, right, and you call these variants, you see that they're biallelically expressing, okay? So we, did the same thing for JAK1, but what ended up happening was the following. Remember, this individual is mosaic, so 50% of the cells should be expressing just the wild type allele. And apologies. So 50% of the cells should be expressing the wild type allele, but the other 50% of the cells should be completely double positive, right? And here we saw them almost exclusively expressing the mutant allele, even though they were heterozygous. So I was staring at this together with Connor, and I'm like, but this makes no sense. I'm like, how can this happen? Where is this coming from, right? So it's really this observation that allowed us to, well, learn a little bit of human genetic inheritance patterns that have been described. I mean, never in disease, frankly, but just as a concept that exists, because monoallelic expression was discovered first in cancer. But then it allowed us really to form a hypothesis that monoallelic expression contributes to phenotypic variability in inborn errors of immunity. So with this hypothesis, what did we do? We took healthy donor blood from 10 New Yorkers, and what we did is we single cell sorted T cells. So we started from a single cell, T cell in each well, and then we expanded it to get these T cell clones. So we would get about 100 to 200,000 cells stemming from one. The idea is that if we sequence two adjacent at RNA level wells, that if their genetics is static, right? But what if one clone commits to expressing of a one allele and the other one doesn't? Then we could prove that monoallelic expression happens naturally in all of us. So we use this method, then pose the question and seeing how ubiquitous is monoallelic expression in healthy individuals. These are healthy donors. So we had this clonal expansion. Now, for us to do this, we of course have to have genotypes. So we have to whole exome sequence and map every single variant. Then we perform bulk RNA sequencing from that 100,000 cells. And then through the computational pipeline, we really merge DNA heterozygosity to the depth of sequencing at RNA level to see if it's 50-50 or deviates from that, which would indicate it's monoallelically expressed. And then we can validate that different ways, either qPCR, digital droplet PCR, and you'll see, I'll show you some examples. So at the time, now there's almost 600 IAI genes. There were 485 PID genes or IAI genes. And of course, for us to be able to assess the gene, that gene needs to have a naturally occurring variation, so SNP, right? And about 3/4 had a SNP at a heterozygous level. And then these genes also needed to be expressed in the T cells, because if they're not expressed in T cells, they're unaccessible in terms of how their transcriptional output is. So we could basically get at about half of the genes that we wanted. Of course, we have data for the whole genome. So this is the computational pipeline that we used. And then I'll show this graph a few times, so I'll take you through it. So on top, you see donors. So this is donor A, D, E, and F. As you see here, 1, 2, 3, 4, 5, those are 5 different T cell clones that have expanded quite well. If something is completely red or something is completely blue, it means it's monoallelically expressing, either maternal or paternal allele, and everything in between is biallelically expressing. The bigger the circle, the more we trust it, because we have higher level of expression and we know that that correlates well, although even with 10, but with 30, we are decently happy transcripts, but often we get many more. So here you can look at donor F, and this is a known imprinted gene, and you can see it's completely red, as it should be. If there is no circles, like in the D here, it means there was no SNP in this gene that was accessible, or it wasn't expressed in that T cell clone. And so this was just a proof of concept that we can detect monoallelically expressed genes that are imprinted. In one individual, they should all be the same if they're imprinted. In every T cell should have exactly the same pattern of expression if it was imprinted, because that's how in the whole organism should be. Okay, and that shouldn't be the case if it's monoallelically expressed, which is now a somatic commitment. So some cells should, some shouldn't have it, although it could be lineage. Okay, so let me orient you here. So we have JAK1 on top, and you can see here donor F. And you can see that in donor F, each of the clones was simply biallelically expressing, as it should be the case. If you look at donor C, however, you can see that some clones were biallelically expressing, but the other ones were biasing towards the alternate allele. Now, if you look at PLC gamma 2 in donor F, you can see that one is alternate allele focused, the other one is reference allele focused, and then 3 other clones are biallelically expressing. So that really showed us that not all donors that are healthy, but some can monoallelically express SNPs that are inconsequential to the function of that protein, because there was no bias in, let's say, missense versus those that were silent variants. This is just the list of the genes. You don't have to read it, but the point is that there's quite a number of genes in healthy New Yorkers here that can undergo monoallelic expression. If we boil that down to all genes, in our study, we showed that about 5% of all accessible genes can undergo monoallelic expression. But that doesn't mean that 5% of my genes will undergo expression. At the individual level, we're usually at about 0.5% in our experimental system. In the context of inborn errors of immunity, it's about 4% of genes that we detected in healthy New Yorkers undergoing monoallelic expression. Okay. So we validated that, and we can validate that, of course, at the DNA level we do Sanger sequencing, but at the RNA level, we take these clones and perform digital droplet and basically get exactly the same result, where some clones are completely committed here to the alternate allele, some are biased towards the alternate allele while still keeping some of the reference allele. This was also true for PLC gamma 2. Again, this is four different clones that looked exactly like when we RNA sequenced them. Perhaps more importantly, this phenomenon appears to be stable for weeks in culture. So at week 1, this is a DNA 50-50, and now at RNA level, we have a bias towards the reference allele at about 94%. But if you look at week 4, we are still extremely biasing towards the reference allele, with the variance being about 10% only. Okay, we wanted to figure out what is regulating monoallelic expression. So here I'm showing you a different plot. This is a digital droplet PCR plot. This is a reference allele for PSC gamma 2, and this is the alternate allele. So you can see that there is a bias here towards the reference allele. So if we give it scramble RNA, nothing happens, you get the same thing. But if we give it JMJ-D3 siRNA, which will alternate the level of H3K27 methylation, we can now make this biallelically expressing. So we know that histone 3, lysine 27 trimethylation can play a role, but it doesn't have to, because in some of the clones, nothing happened. And in the others, we could really see a nice shift. And similar pattern is true now. We have PLC gamma 2 that is biallelically expressing a different T cell clone. But when we do siRNA for DNMT1, which will regulate DNA methylation, we can make it monoallelically expressing. So we know that DNA methylation and histone methylation play a role as well. We tried playing with acetylation and H3K4 trimethylation, but we didn't see any shifts there. So we're still determining if that's the case. Okay. So back to JAK1, which is what, as you remember, inspired all of this study with this genetically mosaic patient, which is how we accidentally discovered monoallelic expression. So JAK1 mediates response to 25 plus different cytokines. And when we initially got this patient that had a mutation S703I, we deemed it gain of function for this reason. If you take a cell line that doesn't have JAK1 and you give it 100 units of interferon, there will be no signaling in terms of phospho-STAT 1 and phospho-STAT 2 because there's no JAK to transmit that signal. If you introduce back wild type allele in cells that don't have JAK1, you can start restoring signaling of phospho-STAT 1 and phospho-STAT 2 in presence of interferon. Now, if you do the same thing, but instead of giving it luciferase, which is a negative control, or wild type JAK1, which would be a positive control, we give the patient mutation. You can see that even at 0 interferon, it was signaling very strongly. So that truly indicated to us that it was an intrinsically gain of function allele. Now, I told you what brought this here, but there was no incomplete penetrance in this family, so we couldn't test that. But we got referral of many, many, many more JAK1 families since we published this now 5 years ago. And here is one family where this is the pedigree. You have a healthy father who carries the mutation, sick mother who carries the mutation, and a healthy daughter who carries the mutation at a position S700N. The mother was actually healthy until the age of 28, and then following toxoplasmosis infection, which of course was and can be relatively easily controlled, she keeps having recurrent infections, CVID, hyper-IgM, splenomegaly, severe pancytopenia, thrombocytopenia requiring transfusion. So all of a sudden, she became very ill. So the first thing we did is just like in the other assay, we cloned this variant, and you can see that it's clearly gain of function. If we performed PCR, If you compare the purple to the black, this is log 2 relative units of a gene that's downstream of JAK1. You can see that it's very clearly gain of function. So we first performed digital droplet PCR, and here, of course, the carrier was completely wild type. And now this was the healthy carrier, which is the daughter. And if we looked at the daughter was biasing towards the wild type allele. She wasn't at 50-50. So she was suppressing the mutated allele. But even the affected carrier wasn't at 50-50. She was also suppressing the mutated allele that we know is pathogenic based on the biochemical assays. So what we did further is we sorted B cells, monocytes, NK cells, and T cells. And here what I'm plotting is JAK1 mutant allele expression. So heterozygous individuals, if they were biallelically expressing, should be hovering here around 50. But you can see that in all cell types, there was this suppression of the mutated allele, as if it was environmental adaptation to something that we don't want, right? With the exception of maternal T cells that were here technically biallelically, but expressing a lot of the mutated allele. So this was the first example where disease segregation segregated with the quantity of the disease causing allele expression. So next thing we did is we looked at PLC gamma 2. As you can know, these patients are plate patients, and I was reading this paper, it was published in New England Journal of Medicine a while ago, and there were these two cousins that had variable disease expressivity. Patient A had normal IgG, and patient B couldn't produce immunoglobulin. So I contacted Josh Milner and Mike Umbrello, and in collaboration with Erica Calzoni in their lab, we took these two cousins PBMCs that they dug out from liquid nitrogen that was there for 10 years, compared it to healthy controls, sorted B cells because we know that there was a phenotype there of this incomplete penetrance or partial disease expressivity, and sorted them based on the flux, calcium flux. So let me orient you here. Again, this is results from digital droplet. This is the wild type PLC gamma 2. This is the mutated PLC gamma 2. The cells can be fluxing or not, and they're either circles or triangles. Healthy donor is blue, so you can see whether the cells are fluxing or not. They're expressing only the wild type allele. If we take patient A, the one that can produce immunoglobulins, whether the cells are fluxing or not, they are biallelically expressing. They are double positive as they should be for each of the copy that they were carrying. However, the patient that had low IgG levels, the cells that were not fluxing exclusively expressed the mutant. So to us, this was, again, a first example that this can be cell type specific, but not just cell type specific. It can be the functional outcome of within the cellular subpopulation. Because if we, mix these cells, the result would be somewhere here. So it would look like a bias as opposed to full-blown commitment to just the faulty allele. Okay, so then we looked at the third family. This was the case of mutations that are in STAT1 that are dominant negative. And the kid was fully ill and the dad didn't have the disease. So what we did is we sorted T cells, B cells, NK cells, and 3 different types of monocytes. To orient you, again, the more droplets that we have, the bigger the circle, the more we trust. If it's in blue, it's going to be completely wild type. If it's in red, it's going to be completely mutant, and everything in between is biallelically expressed. So now, if you look at healthy control, everything is blue, as it should be, because it's only wild type allele that is expressed in healthy control. However, unaffected carrier here, dad, only expresses the wild type allele. We couldn't find the mutant allele in the healthy father, even though genetically it was there. And even the sick carrier It was only in classic monocytes and B cells that it was biallelically expressing. Even the sick boy in T cells, NK cells, and different type of intermediate monocytes was expressing only the wild type allele. So in conclusion, perhaps for me, the biggest question is why does monoallelic expression occur? As I told you in the context of 10 healthy New Yorkers, I don't have the answer. We're trying to work it out. Um, Hopefully, I've shown experimentally that monoallelic expression renders explanation to some incomplete penetrance cases. We are getting quite a few referrals now, and I can tell you quite a bit more is coming. So I have no idea if this is going to explain, 5 or 10% of incomplete penetrant cases or 50. I don't know. We'll just have to keep testing. And then from genetic point of view, in terms of both disease management and diagnosis, we had to make up a word which we called transcriptotype, because in case of PLC gamma 2, disease is fully penetrant only in transcriptionally homozygous state. So just, if you're haplo insufficient, you have a disease. But if you stop expressing the wild type allele, then you have what a full-blown disease is, right? And then due to MA, genetics suggests dominant disease, transcriptotype not manifesting if mutant allele is suppressed. This was the case in JAK1. So now you can imagine caring for a patient family where somebody is not ill yet, but maybe we can monitor the levels of their transcript over time and intervene before disease starts, if there are tools to do that. So I'll stop here and happily take any questions.
Thank you very much, Dushan, for your talk. We have two options for you to make questions. You can either write your question in the chat webinar, or we have the question and answer session here. So we still don't have any question, but I'm sure they're all thinking about everything that you told us, and thinking about many families that they have already followed in their clinics or even in the lab.
Sure.
And of course, this is not the only way to manifest with the incomplete penetrance, but I think it's a very important one. And I would like to talk to you about a family that I'm following in the clinics. So the baby, like the family has a genetic disease. It's not primary immunodeficiency.
Sure.
So the mother decided to take the genetic test. Here we have the first day genetic screening test.
Sure. Yeah, newborn screen, yeah.
Yeah, it's not newborn screening, like they can pay. It's not
Okay.
universal, but they have like some genes that are important. Many genes for inborn errors of metabolism, but also some
Sure.
genes
Sure.
for immunodeficiency. And then this baby, like a completely asymptomatic baby, was not positive for the genetic disease that had already been diagnosed in the family, but he presented a pathogenic mutation in STAT3 gene.
Hmm.
Well, they came to my clinic, and then I said, well, like we have to, follow the baby and to understand how it, because like there were no other cases in the family.
Sure.
But then I asked the mother, the brother, she has two brothers, and the father to take the genetic test so that we
Sure.
could look for potential bone marrow donors in case
Sure.
we decide for a transplant. And the mother has the mutation, and one of the brothers has the mutation as well.
Yeah. Okay.
So they do not have hyper IgE,
Okay. So none of that. Okay.
but like we know that we can
Yeah.
have
I mean,
hyper IgE without hyper IgE, right? But
yeah.
they are like really, really like very mild symptoms for the mother with like sinusitis, recurrent sinusitis.
Yeah, yeah,
but
yeah.
not the classical
No, yeah,
dominant negative STAT3, you know?
sure, which is much more violent.
Exactly.
Yeah. So I mean, that's a very cool case. I actually think, I mean, you should publish that alone because I think it's important to put to light how many patients who have a known pathogenic variant are not sick, right? So
Hmm.
this family, we could test if they're suppressing the wild type allele. Why
Yeah.
not? Maybe we are, then we know why they're not sick, and that would be great. And then, of course, second hypothesis, and there is few papers coming out now. There's a paper in Jack's Med recently that talked about protective alleles in STING in individuals who have known COPA causing mutations. So it ends up being a digenic inheritance that is protective when you have the pathogenic allele. So that's another way to think about it. Of course, that's more exploratory, and I don't know how there's no
Yeah.
simple test for that.
We
So
also have that case that we published showing like the presence of autoantibodies that,
yes,
you know,
that
like
can
the
rescue the
innate
genetic defect. Yes,
rescue.
yes,
Yes,
exactly.
exactly. Yeah.
Yeah, yeah, the TIRAP mutation, exactly.
Yeah.
Yeah. Yeah, this is really
So,
cool. Yeah. So if you have any of those cases, I mean, feel free to send us, and then we can make a plan, and
yeah.
even exclude that it's not monoallelic expression. I think that's important as well. So then we can look for whatever it is that's protecting this family. I'm glad they're okay.
Yeah.
That's the most
And,
important thing, right?
and you
So,
know, like the baby is now like 4 months old,
uh-huh.
and
And
she had a skin infection, and the culture was positive for Staphylococcus aureus.
um,
But it was a very like mild one in her
well,
fingers.
so
So
okay, so
I don't know. But this is something that I really would like
yeah, yeah,
to
yeah.
talk more about that with you, and if we can study it together.
Sure, absolutely. It would be my pleasure.
And you know, we have a comment here. Carmen Bonfim, she is the queen of bone marrow transplant here in Brazil and
Mhm.
Latin America as well, right, Carmen? She's now in the US. And she says that it might be so difficult to select potential donors for
Yeah.
hematopoietic stem cell transplantation if there is incomplete penetrance.
It is,
Always knew that,
yeah, and
right?
actually, I spoke to a couple of transplanters. I mean, in general, they do not like to transplant heterozygous donors if it's a recessive disease. They shy away. They usually look for wild type donors, and maybe this is the case because maybe the monoallelic thing kicks in, and then you don't have a full rescue of the disease.
Yeah. So it's getting
Yeah.
more and more like the more we know, the more
More complicated.
complicated
I
it gets,
know.
right?
I mean, who to me also, I always thought the genetics is, you get genetic diagnosis, that's it. But now we are suggesting that you need monitoring, like thyroid, right? Every year, let's see where your transcriptotide is. Is it moving? Is it not moving, right? It's wild, yeah.
Yeah. So we have a second question here. It's
Okay.
from Nideshda. She says that she have a patient with JAK1 mutation in C2128CG.
Mhm.
So in protein, it's leucine 710 valine. And she developed a recurrent diarrhea every month with arthritis and rash and an
Mm.
episode of aseptic meningitis. The
Mm.
mother has the same mutation, but she's asymptomatic. I
Yeah.
was wondering if we could do an assay in STAT1, as you show. The patient responded with immunomodulation with baricitinib and is now asymptomatic.
Great. Yeah, we can and we should. Happy to test. It's in the pseudokinase domain, so almost for sure this smells like a true gain of function. And our first patient had huge GI problems with eosinophilic infiltrates. I don't know if this one did, and couldn't hold any food. And then fully responded to JAK inhibition. So she's on the right drug. She's managed well, and So I'm glad to hear that, but happy to test the allele and happy to test that mother is perhaps suppressing the mutated allele.
Yeah.
Yeah.
Yes, it makes us think about
Yeah, yeah, yeah.
so many challenges that we have to face, right? Let me see if we have any other questions. So no questions here. So no one has any other question? They're all scared with your talk. Like, they won't be able to sleep tonight thinking about how to select donors for bone marrow transplantation, or how to deal with the patients. And I think that now we are starting a new era, for example, with this baby that I saw in the clinics, like a really, like, in the first month of life we have a mutation in STAT3, but we still don't have symptoms. We know that many of primary immunodeficiency will have manifestations like later,
Yeah.
and then we don't know exactly how to deal with this.
When to intervene, when to start. Yeah,
Exactly.
not easy, not easy.
Yes. So this webinar, all our webinars, they are recorded, and we have this in our LASID webpage.
Great.
So I'm pretty sure that it will be very interesting for us to watch your talk here, and many others that could not be here will be able to see that. And maybe you're gonna have some questions
Oh,
later
please,
from
yeah,
classic
feel free.
members.
Yeah, yeah, it's easy to find me. Just shoot me an email. Happy to answer.
So Dusan, thank you very much.
Thank you.
And
Muchas
so
gracias. Obrigado.
you're very welcome. Thank
Bye.
you. So we still have a few announcements here. Um, so don't forget to register for our LASID meeting. We are pretty close. It will be held in Buenos Aires in October. Once again, to remind you about the call for clinical cases. It's very important that you submit clinical cases so that it will be discussed during the educational day. It's a great opportunity for you to have your difficult cases discussed there. Our Journal of Human Immunology, please consider the Journal of Human Immunology to submit your papers, and also take some time to explore not the inaugural issue, but soon we're going to have the second one. Another reminding you to join LASID, either as a regular member or the junior member if you're below 35 years old or still in immunology training. And I'm pretty sure that we have a new announcement here that Alfonso told me that he would, the registry, it's back online. So go there and check the numbers. And Alfonso told me that he would add another announcement for the end of our webinar. So maybe he didn't have time to make a new slide, but it was an announcement about the membership fee. So please, if you did not pay the fee, remember to pay the annual fee for the LASID members so that you can pay less for the LASID meeting. So thank you very much for your participation here today. I hope everyone enjoyed a lot. And we look forward to see you tomorrow. We have a webinar with Jacinta tomorrow. So if you go to the LASID website or the LASID Instagram page, you have the information there and you can have access to the link. And next month we'll be back with the webinar here as well. So thank you very much, and I hope you all have a very good night.